Hello students, welcome to EPG Pathshala. I am Dr. Sudhir Verma from Department of Zoology, Deen Dayal Upadhyay College, University of Delhi. Today we are going to talk on module Allergy and Hypersensitivity 1 from the paper Immunology. Now the objectives of this module are, we'll start with the introduction part where we'll see what are the basic concepts of allergy and anaphylaxis. Followed by that, we'll move on to what hypersensitivity is. Then we'll see how these hypersensitive reactions can be classified as the immediate hypersensitivity or the delayed type hypersensitivity. Followed by that, we'll move on to the classification part as given by the Gall and Coombe, where we classified all types of hypersensitive reactions in 4 basic categories as type 1, type 2, type 3 and type 4. We'll start with type 1 hypersensitivity in this module, where we'll see what is the degranulation process of various mast cells and the basophils. What are the mechanisms of the degranulations? We'll see what are the mediator molecules which actually cause the effect of type 1 hypersensitivity. Clinical manifestations of type 1 hypersensitivity, which will include all the disorders and the diseases which are being caused by these type 1 hypersensitive reactions. After that, we'll see how we can actually regulate these type 1 hypersensitive reactions. Various medical tests are available to test or to detect the type 1 hypersensitivity. Followed by that, we'll see what are the possible therapies or the precautionary mechanisms which we need to take to avoid these type 1 hypersensitive reactions. Let us start with the concept of allergy first. We all have heard this term on daily basis basically. The concept of allergy was given by von Parquet. He was a pediatrician. In 1906, he used this term in the classical article entitled as Allergy. According to Parquet, an antigen causes changes in the reactivity, which can be the protective one, which we call as immunity, or it can be a very harmful effect, which we call as hypersensitivity. Another scientist, Rickett and Portier, proposed a different term which we call as anaphylaxis. It is in contrast to prophylaxis for the overreaction of the immune system. Now, for a long time this contradiction was going on between the term allergy and the anaphylaxis. Coca and Cook, they proposed a general term which we call as hypersensitivity to resolve this contradiction between the allergy and anaphylaxis. Hypersensitivity is a general term which was proposed by Coca and Cook. It actually refers to inappropriate, deleterious immune responses raised against an allergen. Another scientist, the Gall and Coombe, proposed a classification in 1963, according to which all types of hypersensitive reactions can be classified in 4 basic types. This classification was based on the type of immune branch which is involved. As we know, there are 2 basic branches, the humoral one and the cell-mediated one. This classification is also based on the time taken to show the symptoms. Based on the duration of showing the symptoms, it can be either the immediate one or the delayed type hypersensitivity. Various immune mechanisms can be used to differentiate these 4 reactions. The cells and the mediator molecules which are involved in 4 different types of classification mechanisms. We'll start with the type 1 hypersensitivity. Type 1 hypersensitivity is IgE based allergic reaction which is raised against various allergens. It is a humoral immunity based immediate type hypersensitive reaction. For example, the asthma. Type 2 hypersensitivity is IgG mediated cytotoxic hypersensitivity which is also humoral immunity based immediate type reaction. For example, the erythroblastosis fetalis. Type 3 hypersensitive reaction is the immune complex mediated, the complex of the antigen and the antibody. Again, it's a humoral and immediate type hypersensitivity. For example, the rheumatoid arthritis. Type 4 hypersensitivity is cell mediated in contrast to RES3. It is cell mediated, it is delayed type hypersensitive reaction. For example, the contact dermatitis. Basic components of type 1 hypersensitivity are allergens, immune cells, cytokines, IgE, Fc receptors for IgE and various mediator molecules. The clinical manifestation of type 1 hypersensitivity includes either the systemic anaphylaxis or the localized anaphylaxis. Various factors regulate type 1 hypersensitive response, which may be the dose and the type of allergen, the process of allergen processing, genetic constitution of the individual, and the relative ratio of Th1 and Th2 cells. Wheel and flare test, radioimmunosorbent assay, and radioallergosorbent assays are the common diagnostic tests which are being performed to test the type 1 hypersensitivity. The treatment and the precaution involves avoidance of allergens. We need to modulate the allergic response, stabilization of the triggering cells, antagonism of mediators, and attacking the chronic inflammations. Various drugs are available in market to cure these type 1 hypersensitive reactions now. The allergy word originated from Greek word allos, which means different or others, and the word argia, which means energy or action, in the sense of change in reactivity or capacity to react. It is a fundamental aspect of clinical immunology. The concept of allergy was given by a Viennese pediatrician, Clemens von Perquet-Freyer, in 1906 in a classic article entitled Allergy. According to the original idea of Perquet, when an individual comes in contact with an antigen, which can be the pollens, the germs, the food particles, etc., a change in reactivity occurs. This change, which was termed as allergy by von Perquet, can induce a protective or harmful response. The protective response makes the individual immune to these antigens and hence called as immunity, which does not manifest any symptoms. On the other hand, there is a harmful response which shows symptoms or signs of contact with antigen and is called as hypersensitivity. The immunity and hypersensitivity can overlap and are a result of same physiological response, that is, the response of host immune system. Unfortunately, this new idea was not easily acceptable due to existing strength of idea of immunity. The revolutionary thought of Perquet came at a time when immunology was establishing as a science. The idea of immunity by notable scientists such as Pasteur, Ehrlich, Machnikov, Baudet, Bering, etc. in 19th century hindered and delayed the acceptance of Perquet's idea. Nobody could even imagine at that time that immune system can harm the host it protects. The French physiologists Charles Riquet and Paul Potier rejected the term allergy as they regarded it as unnecessary new term in addition to anaphylaxis that they already coined. These two scientists were suggested by Prince to study the toxic properties of Physalia, which is a jellyfish. They experimented with the use of isolated jellyfish toxins as vaccines. They injected dogs with purified toxins, followed by a booster. Instead of producing antibodies against the toxin, the dog immediately showed vomiting, diarrhea, asphyxia, and even death. These dogs clearly overreacted to the antigens, which was termed as anaphylaxis by Potier and Riquet, in contrast to prophylaxis to describe this overreaction. Riquet was awarded the Nobel Prize in Physiology or Medicine in 1913 in recognition of his work on anaphylaxis. Von Perquet and Riquet's perspectives were different, but since anaphylaxis was easier to understand, it gained more interest. The phenomena of allergy was much broader, but the functioning of immune system and its involvement in diseases than anaphylaxis, an initial purely experimental phenomena. During the search for active substances of anaphylaxis, it was realized that it is not only the effect of toxin, but some principle in the organism itself is also involved. Sir Henry Dale had major contribution in the elucidation of the same. He, along with the co-worker P.P. Laidlaw, studied the physiological aspects of histamine and developed the concept of histamine shock as basis of anaphylaxis. Arthur F. Coca and Robert A. Cook published many articles in 1923 to 1926 to resolve the allergy and anaphylaxis contradictions. They preferred to employ the word hypersensitiveness as a general term. They divided hypersensitivity into normal type, that is, the contact dermatitis, serum sickness, etc., and abnormal type, that is anaphylaxis, atopy, etc. Later, medical community began to use allergy for human perspective and anaphylaxis for experimental animals. There were no significant modifications in understanding of idea of allergy on its use until 1963, when Philip George Gell and Robert Royston Coombs classified hypersensitive reactions in their book Clinical Aspects of Immunology. The primary aim of an immune response is to remove the exogenous or endogenous antigens by various mechanisms using effector molecules. The body homeostasis is regained after a localized inflammation and removal of antigen without causing a damage to host tissue. But under certain circumstances, this beneficial inflammatory response can cause deleterious outcomes, including host tissue injury, diseases, or even death. Such a harmful, inappropriate immune response is termed as hypersensitive response or reaction, or simply the hypersensitivity. The hypersensitive word implies that a person is excessively or more sensitive to an antigen as compared to normal response shown by other persons. Though it indicates towards an elevated or increased immune response, it is not actually the case always. An appropriate immune response is also considered as hypersensitive besides a heightened immune response. Let us now discuss the difference between an immediate and a delayed type hypersensitivity. As we said, based on the time taken to manifest the symptoms, a hypersensitive reaction can be either the immediate one or the delayed type. If the symptoms are observed within minutes or hours of antigen encounter, it is called as immediate hypersensitive reaction. The anaphylactic reaction initiated by antibody or antigen-antibody complex, that is humoral immunity, come under this category. The other category, which we call as delayed type hypersensitivity, is named so because of delay in appearance of symptoms until several days after exposure. Hypersensitive reaction in the course of cell-mediated immune response come under this category. Let us discuss the classification given by Gell and Coombs for classifying the hypersensitive reactions. Based on the type of immune response involved, that is, the humoral or the cell-mediated, and the effector molecules involved, different immune mechanisms are involved in generation of hypersensitive reactions. Accordingly, hypersensitive reactions can be categorized in different types. The British immunologist Philip George Gell and Robert Royston Coombs proposed a classification scheme for hypersensitive reactions in their book Classical Aspects of Immunology in 1963. They divided hypersensitive reactions as type 1, type 2, type 3 and type 4 hypersensitive reactions. The classification is based on the type of immune branch involved, that is, either humoral or cell-mediated, time taken to show the symptoms, that is, the immediate or delayed type hypersensitivity, immune mechanisms, cells, and mediator molecules involved. Here you can visualize a pictorial representation of different hypersensitive reactions. Let us move on to type 1 hypersensitivity now. Let us discuss the general aspects of type 1 hypersensitivity. Type 1 hypersensitivity is an immediate type hypersensitivity within the humoral branch of immunity. It is induced by antigens called as allergens. Type 1 hypersensitivity is similar to normal humoral immune response, except the fact that plasma cells secrete IgE antibody in response to activation of allergen-specific Th2 cells. The IgE antibodies bind with high affinity to Fc receptors present on blood basophils and tissue mast cells. These IgE-coated mast cells and basophils are called as sensitized cells. The exposure of same allergen later causes cross-linkage of these membrane-bound IgE on sensitized mast cells and the basophils, causing their degranulation. These granules release pharmacologically active mediator molecules, that is, the histamines, which primarily cause vasodilation and smooth muscle contractions in a very localized manner or maybe the systematically. The clinical manifestations of type 1 hypersensitivity includes systemic anaphylaxis, or localized anaphylaxis, including hay fever, hives, eczema, asthma, food allergy, etc. A general type 1 hypersensitive reaction is shown here. Let us discuss the various components which are involved in type 1 hypersensitive reaction. Allergens. The term allergen refers to non-parasitic environmental antigens which stimulate type 1 hypersensitive response in allergic or atopic individuals on repeated exposures. The significantly high serum IgE level is mounted in humans against parasitic infections only, but the people who are genetically predisposed to development of IgE-mediated immediate hypersensitivity, that is type 1, against common environmental antigens have this abnormality called as atopy. These individuals have regulatory defects for IgE, because of which non-parasitic antigens also stimulate IgE production, causing tissue damage and hypersensitivity. The inherited atopy phenomena is multigenic. The genetic cause behind the phenomena of atopy has been mapped to various loci by linkage analysis, which are: on chromosome 5 is present a locus that is linked to a region that codes cytokines IL-3, IL-4, IL-5, IL-9, and IL-13, granulocyte monocyte colony stimulating factor. All these cytokines promote IgE production. Additionally, on chromosome number 11 is present another locus that is linked to a region that encodes the beta chain of high affinity IgE receptor. On chromosome number 6 is present the MHC genes, which have also been linked to IgE response. Here you can see some of the very common allergens, which can be the food products, the insect products, drugs, plant pollens, proteins, and various other. Most of the allergens are small-sized, soluble in nature, glycosylated proteins or protein-bound substances with molecular weight of 15,000 to 40,000. The allergenicity of an allergen depends on its ability to evoke a Th2-mediated immune response, which in turn depends on number of factors such as type of allergen, dose of allergen, sensitization route, adjuvant, recipient's genetic or hereditary predisposition, that is atopy. Another component of type 1 hypersensitivity is Th2 cells. The type of immune response against a particular antigenic challenge is determined by the cytokine secretion patterns of different subtypes of Th or T helper cells. CD4 positive T helper cells exert their functions through cytokines in autocrine or paracrine way. There are two subtypes of CD4 positive T helper cells, that is Th1 and Th2 cells. These two subsets differ in the profile of cytokines they secrete. Th2 cells secrete GM-CSF, interleukins 3, 4, 5, 10, and 13. The primary functions of Th2 cells include IgE production, eosinophil and mast cell activation and differentiation, and help to B cells. Interleukin 4 secreted by Th2 cells promote the class switching in B cells from IgM to IgE. This effect on IgE production messes up with the differentiation and activation of eosinophil by IL-5 because eosinophil possess Fc epsilon receptors which bind IgE. The parasitic infections and allergic reactions induce Th2 responses leading to antiparasitic and type 1 hypersensitive reactions. The Th2 cytokine profile is higher in allergic diseases and helminthic infections. Let's move on to another component, that is immunoglobulin E. Immunoglobulin E or IgE is named so in reference to E antigen of ragweed pollen, which is a potent inducer of this class of antibody. Though the amount of IgE in serum is very low, but its potent biological activity made it to be identified. It was identified by Otto Karl Willi Prosnitz and Heinz Kastner in 1921 as a serum component responsible for allergic reactions. They injected serum from an allergic person to a non-allergic person intradermally. The later injection of appropriate antigen at the same site caused swelling and redness termed as wheal and flare reaction. This reaction is called as PK reaction after the name of their discoverers, Prosness and Kastner. It was the first evidence of biological activity of IgE, though the IgE was not known at that time. The IgE was first identified by Kimshige and Teruka Ishizaka in 1966. They immunized the rabbit with serum of an allergic human individual to prepare anti-isotype antiserum. The serum of rabbit thus obtained was reacted with all the known antibodies at that time. This way, each of the anti-isotype known antibodies was precipitated and removed from the rabbit antisera. The remaining serum was still having an antibody which could block the PK reaction, and this new class of antibody was named as IgE. IgE mediates two immune functions, that is, allergic reactions or type 1 hypersensitive reactions and antibody-dependent cell-mediated cytotoxicity antiparasitic defense. IgE has higher molecular weight than IgG due to additional constant region domain which replaces the hinge region of IgG. Here you can visualize some of the properties of human serum immunoglobulin E. It has molecular weight of 1,90,000 as compared to 1,50,000 of IgG. The heavy chain component is epsilon. The light chain component can be either kappa or lambda. The normal serum level of this particular antibody is 0.1 to 0.4 microgram per ml. The in vivo serum half-life is around 2.5 days, and the primary function involves mast cell and eosinophil degranulation. The mast cells can be activated by a number of stimuli, which then can release a spectrum of mediators in discriminating manner. These mediators include histamines, protease, and other enzymes, growth factors, chemokines, cytokines, etc. Mast cells adapt various phenotypes based on their microenvironments, and this property is called as mast cell heterogeneity. Rodents have two major subtypes of mast cells, that is, mucosal type and the connective tissue types. The differences between the two are shown here. Basophils are the non-phagocytic granulocytic leukocytes that are found in most vertebrates. They are the least abundant population of granulocyte in peripheral blood, comprising 0.5 to 1% of total leukocytes. The primary function is release of histamines and leukotrienes in response to IgE-mediated activation, that is, role in type 1 hypersensitivity. The biological activity of IgE depends upon its binding with its receptors. Two types of epsilon receptors are there based on their differences in affinity for IgE, cellular distribution, and their effects post-IgE binding, that is, Fc epsilon R1 and Fc epsilon R2. The type 1 receptors is structurally composed of 4 polypeptide chains, and it is high affinity IgE receptor, whereas the type 2 receptor is structurally composed of single polypeptide chain and is low affinity IgE receptor. Degranulation is simply a cellular process of releasing bioactive molecules from cytoplasmic granules, secretory vesicles in cells like mast cells and the granulocytes. Various mechanisms, including the ones that can bypass the need of allergen or IgE, are shown here in the diagram. A series of well-coordinated intracellular events help in activation and degranulation or release of mediators from the mast cells or the basophils. Various proteins, lipid kinases, and phosphatases along with cytoskeletal rearrangements are involved in these biochemical events. A sequential mechanism along with the pictorial depiction of the same is elaborated here. The mediators are molecules which are pharmacologically active agents that are released from the mast cells and the basophils in the process of degranulation. Based on the synthesis and the storage, these mediators are classified as primary or secondary mediators, shown here with their biological activity. The clinical manifestations of type 1 hypersensitive reactions are classified as localized anaphylaxis, for example eczema, asthma, food allergy and rhinitis, or the systemic anaphylaxis depending on the extent of tissue or the organ damage, whether it is localized to target site or systemic in nature. Here you can see a pictorial representation of various type 1 hypersensitive clinical manifestations. Let's discuss the regulation of type 1 hypersensitivity now. The allergenicity of an allergen, or in turn the level of IgE response by an allergen, depends upon various factors such as the dose of allergen, the genetic constitution of individual, mode of allergen presentation, and the relative levels of T helper 1 and T helper 2 cell subtypes. Let's move on to the detection of type 1 hypersensitive reactions. Identification and assessment of type 1 hypersensitivity can be done by a number of medical tests, which can be the wheel and flare test, which is the simplest, inexpensive and easy testing mechanism for type 1 reaction. It is a skin testing mechanism where an intradermal injection or superficial scratching of potential allergen is applied on specific skin sites. If an individual is allergic to that particular antigen, localized wheel and flare reactions is observed within 30 minutes. Other methods which can be utilized for detection of type 1 hypersensitivity can be the radioimmunosorbent test and the radioallergosorbent test, which are quite expensive but highly sensitive as compared to wheel and flare test. Let us now discuss the therapies or precautions for type 1 hypersensitivity. Starting from the exposure of allergen to reproduction of atopic diseases, there are several points which can be targeted for therapy of type 1 hypersensitivity. These points can be the avoidance of allergen, the modulation of immunological response, stabilization of the triggering cells, antagonism of mediators, and attacking the chronic inflammation. A large number of drugs are now available which can be used for therapy or treatment of type 1 hypersensitive reactions, such as the antihistamines, cromolyn sodium, beta antagonists, cortisone, etc. The events and the possible treatments approach during atopic allergies are shown here in the diagram. So students, let us now summarize what we have learned in this module. We started with the concept of allergy and anaphylaxis. We read that concept of allergy was given by von Perquet in 1906, which refers to overall response of an individual to an antigen. If response is protective in nature, it is called as immunity, whereas the harmful deleterious effect due to inappropriate immune response is termed as hypersensitivity. Another term for similar response was proposed by Rickett and Portier to denote the overreactive immune response. Crook and Coca proposed a generalized term which we now know as hypersensitivity, which was classified by Gall and Koob in 4 different types of hypersensitivity based on the immune response involved, time taken to show the symptoms, effector mechanism, etc. etc. Type 1 hypersensitivity is IgE based, allergic reaction raised against allergens. It is humoral type and immediate type hypersensitive reaction. The examples include the asthma. Type 1 hypersensitivity is the type 2 hypersensitivity is IgG mediated cytotoxic hypersensitivity, which is also a humoral type, and the immediate type hypersensitive reaction. Type 2 hypersensitive reaction can have the example like erythroblastosis fetalis. Type 3 hypersensitivity is immune complex mediated, humoral and immediate type hypersensitivity. The examples may include the rheumatoid arthritis. Type 4 hypersensitivity is cell-mediated hypersensitivity. It is a cell-mediated immunity-based, delayed type hypersensitive reaction. The examples is contact dermatitis. Type 1 hypersensitivity is an immediate type hypersensitivity which is caused by a number of allergens. It is similar to normal immune response except the fact that the plasma cells secrete IgE antibody in response to activation of allergen-specific Th2 cells. The components include allergens, which can be airborne, foodborne, or the drugs, immune cells, which can be the T helper cells, mast cells, basophils, B cells, etc., various cytokines such as IL-4, IL-5, IL-3, etc., IgE, Fc receptors for IgE, and the various mediator molecules. The degranulation of basophils and mast cell is the key point in type 1 hypersensitivity reactions. The generalized mechanism of type 1 hypersensitive reaction involves sensitization and the degranulation phase. Sensitization phase involves exposure to allergen, activation of Th2 cells, release of IgE from plasma cells, and binding of these IgE with Fc receptors on mast cells. Degranulation phase involves the release of the mediators on re-exposure of same allergen and downstream effector functions. Clinical manifestation of type 1 hypersensitivity includes systemic anaphylaxis and the localized anaphylaxis such as allergic dermatitis, hay fever, asthma, etc. Various factors regulate type 1 hypersensitive reactions such as the dose and the type of allergen, processing of the allergen, genetic constitution of the individual, and the relative ratio of T helper type 1 and type 2 subtypes. Various tests such as Whelan flare test, radioimmunosorbent assays, and the radioallergic sorbent assays are common medical tests to detect the type 1 hypersensitive individuals for a particular allergen. As far as the treatment or the precaution are concerned, it can be the avoidance of allergen itself, the modulation of an immunological response, stabilization of the triggering cells, antagonism of mediators, and attacking the chronic inflammation, for which a number of drugs are available to cure these hypersensitive reactions.